completed his PhD study in genetics at UC Davis. He is currently the director of Arizona Genomics Institute and a chair professor of the School of Plant Sciences, Ecology, and Evolutionary Biology at the University of Arizona. Ladies and gentlemen, please welcome Dr. Rodwing.
Good afternoon, everybody. Thanks for showing up. This is a huge room, so it'd be nice to be a little bit smaller and more intimate. But anyway, we have, I think, an exciting program for you today. We have 4 talks, and then 4 20-minute talks, and then 4 genome sequence updates. Kishore Jena could not make it to the meeting today. He was called in sick, and because of my talk yesterday on the feeding the 9 billion people and more of a policy talk, I'm going to go ahead and give a talk in his place, and then after that we'll have Ryuji Ishikawa give the next talk. Okay? All right, the title of my talk today is Sequencing the Collective Oryza Genome to Help Solve the 9 Billion People Question. We talked about the 9 billion people question yesterday, so I won't get into that too much. But the main point here is that I'm going to discuss with you today is the Rice 2020 idea was to essentially determine the function of all genes in rice. And one of the aspects of that is to understand the wild relatives of rice. This is a slide depicting a number of the wild relatives, and the point here is that the wild relatives are a virtually untapped reservoir of genetic variation that can be used to improve cultivated rice. This is a phylogenetic tree of the genus. The genus is about 15 to 20 million years in age, and the main point here is that we have 23 species, wide distribution in habitats, 10 distinct genome types, and 3.6 genome size variation. The main primary gene pool for the Oryza are the AA genomes, and that includes sativa, glaberrima, the 2 cultivated species, Navarra rufipogon, Barthy, meridionalis, longistemmata, and Oryza glaberrima. This slide just shows some of the traits that can be identified in some of these species. We've had a focus on some of the African species, and they are essentially a treasure trove of traits. There's a number of traits involved in drought tolerance, high mineral content in soils, and a number of biotic stress resistances from bacterial blight to Tungaro to the brown planthopper, and we heard about some of the importance of the brown planthopper today. We started a consortium called the Oryza Map Alignment Project, and it has now led into the Oryza Genome Evolution Project. The majority of the work has been funded by the National Science Foundation, and the goal of the project is to create a genome-level closed experimental system that can be used as a research platform to study a number of different topics, from basic research to applied breeding. This is a list of some of the objectives of the 2 projects. There were a lot of resources that have been developed, and then these resources have been analyzed in great detail. They include 18 back libraries representing all Oryza genome types, 18 physical maps, 15 chromosome 3 short arms. Chromosome 3 was one of the chromosomes that we were involved in sequencing during the International Rice Genome Sequencing Project, so we have a great interest in that. Also the generation of 4 reference genomes, Oryza glaberrima, or Barthy, punctata, and now Liercia, which is the outgroup for the genus Oryza. 36 baseline RNA-seq small RNA data sets, and a number of population data sets from the AA genome species. Everything in yellow essentially has been completed, so all of these resources have been generated, and the majority of them have been deposited in public databases. We've done a tremendous amount of analysis and data presentation. A lot of the data can be found at grameen.org and omap.org. One of the main outputs of the OMAP project was, again, a set of physical maps that were aligned to the Oryza genome, to the reference genome. What you can see here, let me find my pointer here. This depicts the chromosome 3, the sequence, and on the right, I can't, the yellow and blue boxes, those represent the physical contigs, and the blue lines represent back-end sequences. And we see these are the AA genomes on the left from Nuvara to Glabrima, and then we go all the way down to Granulata, which is the GG genome. The main point here is that we see a tremendous amount of collinearity, but we also see a lot of significant structural variation. So we've analyzed this data set in great detail, including the back-end sequences, the physical maps, and a number of other resources. And we and others have come to the conclusion that a single reference genome for the genus Oryza is insufficient to capture the allelic diversity hidden within the genus. And we've heard many talks today and yesterday about a lot of resequencing of a lot of the cultivated species and some of the putative progenitors of sativa. But there is also a lot of other variation that is identified in the more distant AA genome species and further down the evolutionary tree. So because of this interest in understanding the genus at a whole level, we've organized a group called the International Oryza Map Alignment Project, and we've had a series of grand challenge meetings that have been held in conjunction with the International Rice Functional Genomics Symposium on Rice Functional Genomics in Tsukuba, Jeju, Manila, Bento Conclaves, Taipei last year, and now in Chiang Mai this year. And kind of the three principal areas that we've been interested in focusing on are, the first is to generate a set of reference genome sequences and transcriptome data sets for all 8 A genome species and a representative of the 9 other genome types. Generate a series of advanced mapping populations, and the identification collection of naturally occurring populations of the wild Oryza species, and for conservation, population, and evolutionary analyses. Today I'd only have time to talk about the reference genomes. This is a current list of the 11 of the genomes of Oryza. Everything in yellow has been is completed or near completion, and Gluma petalata and Meridionalis, the final 2 AA genome species are in assembly or under sequence now. The species that are underlined will be updated today in the second half of the IOMAP session. On the right are the people that have been involved in these sequencing projects. We have the IRGSP gold standard. We have the Indica genome 9311. Yui Singh from Taiwan is leading the Nuvara sequencing project. Bin Han, the Rufupogon, Bin Han and Nori Karada. Our lab has been involved in Glabrima and Barthi. Wen Wang's group has been involved in the Longa Seminata. Olivier Pinot, Antonio Oliveira on the Gluma petula genome. Our lab was also involved in Punctata, Minxing Chen on Brachyantha, Olivia Pinot and Robert Henry with Meridionalis, and then our lab has been involved in sequencing the outgroup. The other 5 species listed here are also under sequencing or being, the sequencing has been initiated. The 3 CC genomes of the Oryza were assigned to Nori Kurata's group, and you'll hear a little bit about that today. Olivier Pinot has agreed to take on the Australiensis genome, which it's about a 1 gig genome, and 60% of it is composed of 3 retrotransposable element families, so that's going to be a tough one to complete. And then Lishi Gao has agreed to work on the granulata genome, and he'll update us next year on that project. Okay, so I'm going to tell you about briefly tell you about the status of glabrema, barthy, punctata, and Liersia. The glabrema genome was the first genome that we assembled ourselves, and we used essentially entirely 454 and Nubler assembly, and then we assembled the genome and then used Oryza synteny to the Oryza to order and orient some of the contigs, and then turn that into an assembly. Your barthy and punctata genomes, we used 454 and Illumina. We used Nubler for the 454 data, AllPaths for the Illumina data, and Minimus 2 to put the 2 data sets together. And then we used some software that we developed called Genome Puzzle Master to help edit the assembly. And finally, Liersia is a complete Illumina AllPaths genome assembly. And the nice thing about all of these resources is that they're all linked to the physical maps, which is something that's very unique compared to a lot of the other genome projects. We're probably one of the only genome projects in the world that has physical maps to back up all of the genome sequences that we're generating. I'll tell you a little bit about glabrema. Glabrema is the other cultivated species of the genus Oryza. It has increased tolerance to biotic, abiotic, and weak competitiveness. The goal was to generate a reference sequence. We used a back pooling approach in next-gen sequencing. So we pooled 30 backs at a time and sequenced them, assembled them, and did that across the genome, and then put that all together. We've generated transcriptome data. We've done baseline annotation and topic-specific annotation. This is a summary of the assembly. It's 316 megabase assembly. We have a contig N50 of 19 kb, scaffold N50 of 216. We had about 5,300 scaffolds, and 90% of the genome could be anchored to the aligned to the sativa genome, and 10% could not. And so this is the glabrema assembly. We've done a baseline annotation of that, and I essentially identified 33,000 genes, and we're in the process of annotating and analyzing this genome sequence in great detail. The barthy was sequenced using Illumina and 454 data. This is a pretty nice assembly. It's again about 316 megabases in size. 308 megabases could be aligned to the Oryza reference gold standard genome, and there's about 7 megs that are unanchored, about 2.4% of the assembly. So this is a decent genome. The N50 scaffold size here is about 25 megs. Punctata, also a pretty decent assembly. This one has a little bit more 454 data in it, and the assembly is, trying to find it here, it's about 400 megs in size, 393 again could be aligned to the sativa genome. We have about 7 megs that are unanchored, and an N50 scaffold size of 31 megs. Liersia is a de novo assembly, and this is kind of more of a I would consider a draft type assembly. We used a number of different fragment and mate paired configurations. We have an N50 contig size of 47 kbs and an N50 scaffold size of 9.7 megs. It's a nice assembly, and right now we're involved in editing this assembly using Genome Puzzle Master and the FPC physical map. Okay, so at present we have, we call this the 9 genomes of the Oryza plus the Ursia. The 4 species in yellow are the ones that we were involved in sequencing. We have all of these genomes available now, and the goal now is to do have a common repeat annotation database for these 10 genomes. We have transcriptome data sets already for all of these genomes, and they've been Trinity assembled, and we've had reference guided assemblies for them. And the final task is to, before we do a deep analysis, is to do a common annotation across all these genomes. And we're using the software called Maker, and I'll tell you a little bit about that in a second. So this is a Maker annotation using the Barthy Brachyantha and punctato assembly. A Brachyantha was provided by Minxing Chen, and you can see that the number of gene models that I've called are pretty consistent across the genomes. There are fewer in Brachyantha, and Brachyantha has the smallest genome of all the Oryza, so that data makes some sense. Right here is everything in yellow is what we have run through the Maker annotation pipeline, and we have 3 more genomes to go, Pogon, Navarro, and Longisteminata, and then we'll have that Maker data set already available. So we're essentially we'll be done with that hopefully by the end of this year, and then we're doing a lot of comparative analysis amongst all these genomes with a number of collaborators. Okay, I'm almost out of time here, so I'll just tell you a quick story. Here we, in addition to the reference genomes, the transcriptome data set, we've also done a number of, generated a lot of population data sets. So I'll tell you a little bit about Oryza Barthy, population structure analysis. This is some work of a graduate student of mine called Mu-Hua Wang. Population structure is the partition of individuals in population based on their genetic variation, and it can be used, can be caused by several factors including selection, drift, migration, and gene flow. And investigation of population structure can help to understand the evolutionary history and diversity of a species. We essentially resequenced 98 Obarthy sessions and ran it through a program called Structure, which allows you to determine population structure. And the results showed that we had essentially 4 subgroups as well as an admixture group within the population. So the 4 subgroups are 1, 2, 3, and 5, and the subgroup 4 is considered more of an admixture. And the groups 1, 2, and 3 were distinct from Glaberrima, whereas the group 5 grouped with Glaberrima, and group 4 was a apparent admixture. We also used principal component analysis and developed a neighbor joining tree, and this data also, independent data, also supported the same type of data as the population structure. We're not using this pointer very well. Here it is. Okay, so CG14 clustered with group number 4. This is the admixture, and then we have the 3 groups that are distinct from Glaberrima right here. So the Glaberrima, Obarthia is the putative progenitor of Oryza glaberrima, and there are 2 theories concerning domestication of Glaberrima. One suggests that there was a single origin of domestication along the upper Niger River, and that's from Portuguese. And there was Harlan also suggested that Glaberrima was selected at several different localities within vast forests and savanna areas. So there's a single domestication site versus one that was independently domesticated around all of Africa. So we essentially grouped the 3 distinct groups, 1, 2, and 3, into one group. We're calling that type 1. Those were distinct from Glaberrima. And then the type 2, which had group 5, which was clustered with Glaberrima, and group 4, which was an admixture. And then we determined, essentially grouped them based on their their origin from Gambia down to Tanzania and some unknown sites. And this data here, we plotted this against, or mapped this to the continent of Africa. And the data that we have here essentially supports that that glabrine was domesticated from a single location near the domestication of origin in West Africa along the Niger River. So I'll just say that that data supports that. We're also interested in getting some additional data to support this conclusion. Okay, with that, I'll just say that I think we're getting close to getting all the diploid Oryza species sequenced, and we now have to move toward the polyploids. There's 4 polyploid species, and that's going to be more of a challenge, but since we have physical maps for all these genomes, that is going to reduce the complexity significantly. So lastly, I'll just say that the wild Oryza species are agronomically inferior, but contain a virtually untapped reservoir of genetic diversity that can be used to help address the 9 billion people question. With that, I'll thank my lab. This is our lab in Arizona, and my 3 group leaders, John Wei Shang, Dave Kuderna, and Yisu Yu, and a cast of collaborators around the world. I'll just leave it with that, and I'll definitely want to thank the National Science Foundation for all of their generous support. Thank you. So I have time for, I'm running the show, so I have time for a couple questions. Any comments, questions?